traditional methods as of now that have been devised for utilizing, protein structure prediction. Then we had the intervention of AI. So once AI came about, we evolved into AlphaFold. AlphaFold was developed by a very talented bunch of researchers based out of DeepMind Google. Uh, DeepMind is uh, a collaborative research work done by researchers from Google. You might have heard of Hassabis. So Dr. Hassabis won the Nobel Prize for physics last year, uh, and he won especially for this particular invention, which is AlphaFold. And AlphaFold has created a revolution in the protein structure prediction industry, because it uses the process is like it uses deep learning. So it uses concepts of deep learning, machine learning, and convolutional neural networks to predict 3D structure proteins from amino acid sequence. So a user just has to pass an amino acid sequence in the form of a FASTA sequence, and it will be able to predict the 3D protein structure. So some of the pros were it was highly accurate, it was really fast. So it literally take minutes to predict protein structures for in a very accurate manner, and it's readily available. So if you just go to the AlphaFold official website, you'll be able to see the AlphaFold server, and you'll be able to assess a lot of proteins, and you'll be able to get assessments results for those assessments. Next, which we are going to talk about in greater length in the course of this module, is ESM fold. So ESM fold is evolutionary scaling method fold. So this was developed by Facebook's Meta AI, and the process is and this has been a revolution in terms of protein structure prediction, and in terms of aiding bioinformatics to predict the disease-causing agent in certain protein structures. So the process was using a transformer-based language model to predict protein structures at really lightning speed. So it's very fast. It's even faster than AlphaFold. If AlphaFold can predict the structure of proteins in minutes, ESM-Fold is very fast and it can predict them in seconds. So some of the pros were it was extremely fast, and it's also a free open source tool, so anyone can access the ESM-Fold metagenomic atlas. And one of the cons is that it's slightly less accurate than AlphaFold, because ESM-Fold takes in just one protein sequence, and it calculates the it calculates the PLDT score, which we will talk about in the subsequent slides. And so hence the accuracy is a little hampered because of that. But nevertheless, it's not the differences between ESM-Fold and AlphaFold is the accuracy is minuscule, so you can overlook it and still rely on ESM-Fold. So yeah, so that is a bit about the experimental, the traditional and the AI-based methods that were used for protein structure prediction determination. Next. Yeah. So now we are going to talk about challenges in experimental techniques. So like I said, whatever I mentioned previously for X-ray crystallography, cryo, EM, and NMR, the challenges were plenty, right? So data quality and availability. So it was really difficult for scientists to integrate with the existing traditional techniques, because the scarcity of high quality label data was the high quality data label data was less, and experimental data sets, especially for protein structure, gene interactions, and drug responses, were limited, and hence there was noisy or biased data set. So errors in sequencing, labeling, or annotation can lead to like a lot of misleading AI predictions. Last but not the least, the data standardization issue. There were so many issues faced by using these traditional methods. Some of them were like different labs were using like various varying protocols. There was no standardized procedure. So every lab had their own um, uh, own way of assessing quality of a certain data set, and of bioinformatics protein structure data set. Hence, the labs use varying protocols. and computational complexity. So high computational requirements were required. So transformer-based models like AlphaFold and ESMFold just came about like very recently, and hence previously ethical privacy and regulatory concerns were overlooked, and people were assessing data sets that randomly without knowing the concerns of the patient or anything. But with newer techniques like AlphaFold and ESMFold, you have a personalized platform for assessing your data. Then, last but not the least, the experimental validation. So you never know whether this is coming from a valid source of experimenting. Like it was very difficult to validate AI generated results. So there was no set standard or fast rule that there was no board that could assess these AI generated results. So there was a lack of explainability. There was no accountability there for anyone to come about saying that, oh, this particular AI predicted disease causing agent that causes cancer is actually this is the protein sequence that causes cancer, which is predicted by this AI model. So there was a chance of bias and generalization. So these were like one of the huge black boxes that were that newer techniques like ESM fold and AlphaFold have been trying to combat and come about with reliable sources which follows ethical and privacy regulations, and entrust the user to be given reliable results regarding their diseases or their sickness. So this was a huge challenge that was taken into account when they were building AlphaFold and ESMFold. Okay, so now we are going to the interesting aspect of today's content, where we are going to talk about how the evolution of protein modeling happened, right? So, so basically, so we had early homology modeling was the first principle that was used for proteins to adopt similar structures. So homology modeling meant like it used experimentally determined structures of homologous proteins to predict unknown protein structures. So like in scientific research, the more unknown the proteins are, the more the more curious a scientific researcher would be to explore that data set. So computational methods were evolved in such a way that it could cater to the needs of the not just the scientific researcher, but also the student who is or the subject who wants to know the underlying causes of his disease. So some of the advantages of early homology modeling was it worked well with homologous structures. So homologous structures mean like when you're comparing two different molecules, we look for the similarity between these two molecules. So you would have heard of ancestral genomes. So we all come from the last unknown common ancestor, which branched into archaeobacteria and cyanobacteria, and that's how humans evolved from. Current day humans are have like a common ancestor from where they have evolved from. So a lot of our genes can be similar to a lot of other living organisms. So in order to do scientific research, like we we had to perform sequence identity, and homology modeling enabled that. And then when we get into the deep intricacies of protein folding, it uses structural motifs and patterns from known protein folds to predict structures of sequences with close homologs. So basically, the method is like it aligns the target sequence against a database of known folds using energy scoring functions of machine learning. So basically, it was based on energy functions. So energy function is nothing but the energy taken by the protein protein to fold in a hexagonal structure or a structure that is shown below, right? So some of the advantages of fold recognition and threading was that it could predict structures for proteins with low sequence similarity. Even with 30% less than 30% identity, it was able to it did not have to be very similar for instead of where homology modeling needed to be at least 50 to 70% similar to the original protein structure. This required only 30% identity, but the limitations were the accuracy took a toll. So since you could pass in random data set and assess the folds, the accuracy was compromised, and accuracy generally depends on the quality of the fold database. So it may not work for novel protein folds present in the existing database. Then we have something called ab initio prediction. So the concept of ab initio prediction is that it predicts protein structures from scratch using physics-based principles. So we would have heard of thermodynamics, right? So thermodynamics has like like uh, latent heat of fusion and thermal energy which is calculated. So we had something called Gauss constant. So all of this underlying principles were used to predict the protein structures using physics-based principles. So what methods it used was like it used energy minimization. So the less the energy taken for the proteins to interact with each other and come about a solution and fold into a protein structure where we can where researchers were more focused on finding the disease causing agent. This prediction method helped them to get into the intricacies of finding the potential disease causing microbe or protein. So the advantages of this model was that it it could model proteins with no known homologs, and it provides fundamental insights to protein folding mechanisms. But the limitations include that it was very computationally expensive, it required significant processing power, and less accurate. It was less accurate than the homology-based methods for large proteins. Next, we have advanced force fields and simulation techniques. So the concept of advanced force fields and simulation techniques was that it improved molecular force fields to refine protein structural prediction. So the method employed techniques like molecular dynamics, which simulates atomic movements over time, and then it had Monte Carlo sampling, which is a unique concept that was brought into the advanced force fields technique, where it used probabilistic methods to explore conformational spaces. So basically, these protein structures, when we are performing Monte Carlo sampling across it, as we all know, we have used Monte Carlo across a lot of data sets, but it cannot be applicable to proteins. So these probabilistic methods were enabled a human being to explore conformational spaces. So conformational spaces are like different rotational degrees of a protein sequence that can be assessed. So we have LNR versions of a certain molecule, right? So as we have all learned in organic chemistry, the stereochemistry of a particular molecule is really looked into consideration. So what it does is that it combines AI and physics-based models. So whatever ab initio does and whatever fold recognition does, it's a combination of both of these two methods function, and so the advantages of that is that it provides more accurate modeling of atomic level interactions. Now, we were talking about proteins, which were a series of molecules, a series of amino acid sequences, right? But this looks into the atomic level interaction. So this is even more minute, and it captures the protein flexibility and dynamic behavior. Some of the limitations were that it was computationally demanding. Simulations can take 2 weeks to months, so it was not viable for commercial purposes. And the accuracy depends on the force field quality and parameterization. So not every time do we, no, it's not always that we get great interactions between these molecules. Seldom we do, right? So it is very difficult to find. It is very time consuming to find those protein sequences that interact with that provide quality force field. Next, we have the deep learning revolution. This is where tools like ESM-fold and AlphaFold can be taken an example for, which is where it leverages neural networks and AI models to learn complex patterns in protein structures. So the method is using deep learning frameworks like AlphaFold and Rosetta Fold. So Rosetta Fold is another transformer model architecture, which is which is able to perform rapid predictions. So advantages of this is that it drastically improves accuracy and speed and can predict protein structures without even relying on experimental data. Democratizes democratize sorry democratizes sorry again democratizes access to protein modeling for biologists and researchers. Uh, some of the limitations were that it required large-scale computation resources. Performance depends on, like, training data availability. Not we don't have much curated data for this, so we are still working on that. So there are a lot of repositories, that medical repositories like PubMed, NIH. All of these resources are great for looking into protein structures. So next we'll move on to the second course module, the sub-module of this series, which is the fundamentals of transformer-based models in bioinformatics. So, before we dive into this, I'm going to talk about uh, how the transformer architecture works. So as you can see in this model, um, the transformer components include the encoder, decoder, and the multi-head attention mechanism, the addition and normalization factor, the MLP, which is multi-layer perceptron, and which is part of the encoder architecture. And the decoder contains the addition and normalization along with the MLPs in succession with multi-head attention. So when you input a sequence and when it goes through it, what happens is that it converts the raw sequence data into dense vector representation. So it goes to positional encoding and it creates a dense vector representation. In bioinformatics, what happens is that amino acid sequence are embedded. So we pass it in terms of embedding representation. And what happens in positional encoding is that since transformers lack recurrence, this step injects order information. So functions like sine and cosine functions are used to encode these positions. So what happens once it passes through the positional encoding is that cosine similarity operation is performed, and you're able to get a protein sequence that goes into the multi-head self-attention. So multi-head self-attention captures relationship between all elements in a sequence. So it captures the relationship between like a protein sequence, like all elements within the protein sequence, and it gives the model to focus on different aspects simultaneously. Finally, we have next we have the addition and normalization feature. So in the addition and normalization feature, you would have heard of the function called ReLU. So ReLU is basically uh, uh, like an activation layer. What happens in the addition normalization layer is that there's a residual connection, and there's a normalization layer that is able to activate certain probabilistic distributions when the input sequence passes through various weight vectors in neural networks. So the advantages of this is that it ensures stability and prevents vanishing gradients. So the problem of gradient, like gradient loss, is combated when we are using an additional normalization layer. And next is that the residual connections help gradient flow in deep networks. So even if there are some residuals. So residuals in protein structure is nothing but the added proteins that amino acid sequence that are present in a protein sequence that goes through this addition and normalization aspect. So this residual connection helps gradient flow in deep networks. Next, we have the feed forward MLP layer, so the multi-layered perceptron layer. So multi-layer perceptron layer is a fully connected layer that refines attention outputs, and it's applied in transformations and non-linearity. So if there are any non-linear protein structure predictions, what it does is that it normalizes it and linearizes it so that it passes to the decoder architecture, and relevant protein structures that with the amino acid sequence are passed to the decoder structure. So this is repeated. This process is repeated multiple times. So what you see is just one iteration of this process, but this process gets repeated multiple times in order for the attention mechanism model to zero down on that particular amino acid sequence that is causing. If you're predicting diseases, then we are zeroing down on the amino acid sequence that causes that particular disease. So the encoder layer is of multiple identical layers. So there can be around 6 to 12 layers. So you would have heard of the architecture BERT, right? BERT has 6 to 12 layers of real world models. 6 to 12 layers of these multiple identical layers of encoders and decoder architecture. So that has caused a huge boost in finding these disease causing protein structures. In bioinformatics, the most common activation function that we come across is not ReLU but GLU, which we will talk about in the next page. And this GLU function is predominantly used in tools like AlphaFold, ESMFold, etc. Okay, so now we will talk about the evolution of transformers in bioinformatics. So in the previous course, we had talked about evolution of transformers in drug discovery, but now specifically for the bioinformatics use case. So this is like a timeline. So in 2018 to 2019, the first adoption of NLP models for protein sequences were introduced. So this was inspired by NLP advancements like BERT, GPT model, and researchers applied self-supervised learning to biological sequence. So researchers thought to themselves that why don't we apply for a medical use case and create a transformer model that is specifically used for bioinformatics. Hence, they came about so many of these ESM tools like ESMFold and AlphaFold. And there came a tool called UniRep, which was using LSTM, so long short-term memory. So what it did was like it had a learned representation of the protein sequence, paving way to the transformer adoption. Then we had a tool called TAPE. So TAPE was a benchmark for deep learning in protein understanding, which used transformers for sequence learning. So in bioinformatics, one of the ways of alignment is called multi-sequence alignment. And multi-sequence alignment is where your reference protein sequence is aligned to a bunch of sequences, and you're trying to map the similarities between the reference molecule to the multi sequence platform. So one of this tool that enabled the automation of this was tape, and tape came into the market in 2019, and it was a revolution in terms of like deep learning in protein understanding, where it used transformers for sequence learning. Then we will in late 2020, models like ProtBERT, ProtT5, and ESM1, mm, which was the simulate like a like the protocol version of ESMFold that came about um, in its early stages, and prototype. I mean, not protocol, prototype. So what these protein language models were trained was, so they were trained on millions of protein sequences. They were treating amino acid sequences like words in a sentence. So these amino acid sequences were considered as words. So like whatever you see in vector embeddings in BERT architecture, the same concept was included when they were designing tools like ProtBERT, ProtT5, and ESM1. And these models captured evolutionary relationships and functional properties without reading multiple sequence alignments. Then we had in late 2021, we had the first prototype of AlphaFold, which is called AlphaFold 2. So they I don't know why they called it AlphaFold 2, but AlphaFold 2 in fact came before AlphaFold. And so this revolutionized the protein structure prediction by integrating attention mechanisms for spatial relationship. So all this while we were looking at proteins at a linear fashion, but now we were looking at protein in a 3D vector fashion. So AlphaFold allowed researchers and even common man people who were curious to know what disease causing agent caused that disease, they were able to view this protein structure in a 3D, 3 dimensional way. And AlphaFold allowed that. And so because of this 3D, 3 dimensional view, we are able to find out at which stereochemistry of that protein sequence a certain drug molecule would have binded, would be perfect to bind to attack on that protein, attach on that protein ligand, and then you know and then become a more effective way of translates to drug discovery, where you are able to find drug agents that could attack on the right uh, position of that protein sequence. So AlphaFold looked into it and created this concept of spatial relationships for protein sequences. ESM2 was also around that stage. Meta AI and Google DeepMind had this, a competition of which protein modeling sequence would be of the best use case for researchers. And so Meta AI came about with ESM2, and that showed how transformers alone could learn evolutionary scale representations of proteins. Then we have large scale sequence modeling. So the first direct multi-sequence alignment free transformer based structure predictor was called protein MPNN. So this was a generative transformer for designing stable protein structures. And BERT architectures like gene BERT, DNA BERT, all of this extended transformers to genomics and regulatory sequence predictions. Previously, it was just to multi-sequence alignment predictions, but now they transformed it to a level of uh, utilizing transformers for genomics and proteomics use cases. So like examples of gene BERT and DNA BERT were can elucidate this modeling. Then we have the generative AI for bioinformatics, which is to present day what we are working on, right? So tools like ProGen2, which was introduced in early 2023, use transformers to generate novel protein sequences with desired functions. So all this while we were able to assess protein structures, but tools like ProGen2 was able to were able to generate new protein sequences that were not found before. So this has become an advanced function of interplay of AI in bioinformatics, where transformers were able to produce novel protein sequences with specific functions. Like say to treat pneumonia, a certain protein can be activated. So a drug molecule needs to attach to that protein what is that ligand, right? So when the drug ligand attaches to the protein sequence, if you're using tools like ProGen2 and RF diffusion, it could predict new novel proteins that can actually be instrumental in finding out how to combat diseases like pneumonia or Alzheimer's. As people are right now, researchers right now are working on Alzheimer's, where they're finding out the reason behind the neurodegenerative disease, and they're finding out which particular protein is actually getting triggered while this neurodegenerative disease is plaguing a lot of human beings across the world. So people are actually finding out which protein sequence or some hidden protein sequences that were not assessed previously. So tools like Progen 2 and RF diffusion were instrumental in finding out these new protein sequences that were hidden under the background all these years. And then tools like AlphaFold. Now, right now we have the third version of AlphaFold, which is expanding into drug target interaction, ligand binding, and cellular modeling. So all of these tools, you can see how the transformer model has actually evolved across just the span of 5 years. So you can see that if anyone is interested in this research, you can see how the learning rate has to be really exponential for you to keep up with the time of what is actually happening in the protein in the bioinformatics space when it comes to intervention of AI. So you need to become like really quick and adaptive learners to embrace new concepts and embrace the applications of new concepts in these transformer models. So next we will dive deep into what is called the GLU function, right? So GLU function is the function that I mentioned previously that was used for like I said for AlphaFold and ESM fold. This was the like quintessential what do you call, like the function, the loss function that was used when calculating I mean activation function, not loss function, sorry, my bad. The activation function that was used in these architectures like ESM fold and AlphaFold. So the underlying function is like this. So it uses this formula called x into phi of x. So phi of x here is the Gaussian cumulative distribution function, where it is represented by half into 1 plus exponential of x by root 2, right? So this phi of x is this e power x by root 2 is the error function that is integral for Gaussian distribution. So with the error function, it will smoothly gauge the input based on its probability under a normal distribution. So usually this is phi of x is usually a normal distribution curve, but um, when you when you introduce an error function like x by root 2, what it does is that it will smoothly get the input. So when the loss function is calculated, we will have number of troughs and lows, right? And so what this activation function does is that it smoothens that curve and normalizes that particular and makes it more smooth. So that the predictions that you get towards the end are way more reliable and way more accurate. So this is kind of like a new concept that was brought in by researchers. Previously, they were using ReLU, which is a rectifier linear unit. So rectifier function was used. So it was basically like phi of x in that case was just like a y equal to x trend line. So in this case they have used an exponential trend line that smoothens the curve and adapts to any kind of input curve. So like say you're passing like in mathematical terms, say you're passing like a hyperbolic function or a parabolic function. So with like it may not even be like y is equal to x square. It could be x square plus x, something like that. So any random function when passed into this particular GLU model, it would be able to smoothen that model. So this was the underlying principle of a GLU function, and this is what they used in the transformer model architectures of ESM fold and AlphaFold. And why is GLU preferred in bioinformatics transformers, right? This is just a mathematical function, but what is the use case of this in bioinformatics research or bioinformatics, like I mean, functions of bioinformatics in transformers? So one of the biggest use cases was the protein DNA sequences required better contextual understanding. And GLU's smooth activation helped transformers learn representations more effectively. So, like longer dependencies in biological data. Uh, so, so like previously there were lot of dependencies of um, in biological data. Now, with the advent of GLU, we have equal dependencies of the model on biological and mathematical data. So, what GLU does is it avoids the sharp cutoff, like I previously mentioned, that ReLU overlooked, and it led to a better gradient flow. So, the concept of dying gradient is actually, uh, the problem of dying gradient is actually tackled by the GLO function. So self-attention layers need stability, and GLU works better with layer norm and attention mechanism. So normalization layer and attention mechanism, are the two underlying. Like I showed you in the architecture. So you have like a normalization function and an attention function. So the combination of these two are not only on encoders but also in decoders. Like you can see two layers of normalization and attention. Uh, so even in a ReLU, even in a GLU function, that's what happens where it works better with this normalization and attention mechanism put together. So combining these two significantly improves the model's performance by stabilizing the distribution of activations within the attention layer, allowing it to focus more effectively on relevant information across input sequence and discarding the outliers or the anomalies. So essentially making the attention mechanism more robust and expressive. So they can really target on the amino acid sequence. If you're looking at a disease causing protein sequence, they can really narrow down, and the attention mechanism can be really nuanced to focus on that particular amino acid sequence that is expressing when it is getting attached to a drug ligand, and this creates a what we call a drug histocompatibility molecule, where the drug ligand and protein ligand, when attached together, we are able to assess the binding affinity, which is another concept that I'll be talking about in the upcoming modules, right? So yeah, now we will be talking about how GLU is different from ReLU. So activation, so the function is like I mentioned in ReLU, it is 0 to x. So it is a linear function, right? So the graph would look like y is equal to x. The f of x is equal to x. That is the formula for a ReLU function. But GLU is x into phi of x being the error function. I mean, phi of x including the error function. So that is one of the underlying difference in the structure of the function. Then the next one is that the computational efficiency. So in a ReLU function, it is simple and it's very computationally efficient. But the downside of GLU sometimes can be that it is computationally heavier, because see, you have like an error function to calculate, you have x by root 2 to calculate, and you have to multiply it, add 1 and multiply it by half. So there is a lot of computational intensity involved when calculating the GLU function. So the gradient problem, in this case, it help in ReLU functions. It helps mitigates the vanishing gradient problem, because your y is, I mean, f of x equal to x is it's a very what to say, it's a very binary kind of output that you get, either 0 or 1. So it helps to mitigate that vanishing gradient problem. But in a GLU function, it to the gradient problem in a different way. It improves the gradient flow, especially in deep transformers, where like you have, this is an example of a deep transformer where you have multiple attention and normalization layers attached together, right? And using a ReLU function in this kind of an architecture will be a no-brainer, because you have multiple outputs coming from multiple attention head, multi-head attention and normalization models. So what this does is that it it improves the gradient flow. So not like every gradient is discarded, but some gradients can be included as well, and it creates a more adaptive and robust system. So it is especially helpful when it comes to transformers. And for specific use cases like AlphaFold and ESM that has a very complex architecture, using a GLO will be the right idea because it can handle protein structure predictions efficiently. So the problem of, like I mentioned, there's something called the dying ReLU problem in neural networks, where ReLU neurons consistently output zero. So, like I said, it's more of like a binary output that you get from ReLU. Uh, when ReLU neurons consistently output zero. So the weight vectors consistently output zero in this case. Um, Whereas in the GLU function, it becomes inactive and it's unable to learn, which can lead to a network's reduced capacity and poor performance. So like that, a ReLU function cannot be viable for like complex protein sequences. So when it comes to bioinformatics, ReLU can be only employed for simple architecture, but when it comes to you know uh, GLU can be used for like multi-sequence alignment, assessing multiple protein sequences, and it outperforms the deep transformer models. So it outperforms the what to say, like the primitive transformer models that used to be there. So like the original BERT architecture, which used to look like this, it outperforms it by multifold. So using GLO activation function is like uh, has been a game changer in terms of predicting protein structures in bioinformatics. So why? So this is how the activation function functions, right? So you have at x greater than zero. I don't want it to sound mathematical, but I don't want it to look like I'm taking a mathematics class today. But I just want to show, let you know how the beauty of how like these activation functions work, right? So like in a protein DNA sequence that requires contextual understanding, GLU smooth activation curve helps transformers learn representations more effectively. So longer dependencies in biological data, like GLU helps in avoiding that particular sharp cutoffs, leading to better gradient flow. So
You know, one question in that
yeah,
JLU, why you divide by root 2
sorry, by
in
root 2, huh?
yeah.
Yeah, that's a good question actually. So as far as I know, I think like like when we have uh, so x can be very minute. It can be even in terms of like 0.005, like it can be in like the fifth form of a decimal format, right? So when you're dividing by root 2, when you're dividing by an irrational number, so and you're creating and moreover you're adding an exponential aspect to that irrational function. An exponential function of an irrational number is actually a rational number. So you will get it in the format of a rational number. So to avoid, so had I used e power x and had I passed any form of irrational number, I'll probably get a very I'll probably get phi of x as zero sometimes, right? So which is why we are dividing it by root 2. That is my my reason of why it is divided by root 2. But that's a good question. I'll definitely do a research on that and let you know. But I think I also have this question. So x by root 2, see, if you do an irrational number divided by an irrational number, and if you put it on an exponential format, won't you, correct me if I'm wrong, an exponential version of an irrational number Won't you get it as a rational number?
I don't know that, but
Yeah,
uh,
so it is basically to cater to every data point. So if you do by root 2, no matter what the whichever aspect of that graph that we are looking into, it will provide an output. And since x is that particular point, so when you're multiplying x with x, you will get even more nuanced values for f of x. So like it is to reduce the errors. So like if I had used phi of x as half of e power x and not 1 plus e power x by root 2, then probably I'll be getting a different activation function, but it might not be it might not be continuous. So yeah, that is one more thing. So this Gaussian cumulative distribution function has to be continuous, and in order to become continuous, it has to cater to every point on the x-axis and create a y equal to f of x function, right? So for that to happen, I think that is why the scientists would have resorted to x by root 2. I hope that answered your question.
it might be just by 2 or something by other number can be.
Sorry.
It might be some only just x by 2 instead of x by root 2.
uh,
Maybe you can look
No,
at it.
but I have looked into how a GLU function works. We can definitely touch base on this post the seminar. I can show it to you. Uh, the formula for a GLU function is this: half into 1 plus e power x by root 2. I also had this doubt, but they have told. I mean, GLU was specifically defined because ReLU gave only binary outputs. Like ReLU just gave 0 or 1. GLU was able to give you 0.03, 0.04, 0.05. So it was able to not just classify it as binary, but it was also able to give you like what is that? Like if you're if you're looking into stages of cancer, and if you're using a deep learning model, and if you're using GLU function, you're not just performing cancer stages in the terms of whether you're detecting cancer or not. That is what you'll get the output as when you're using a ReLU function. But when you're using a GLU function, you can classify this data set into different stages of cancer. So based on the data points that you feed, it is able to classify whether on 5 stages of cancer, like from benign to malignant, it is able to define that, oh, this particular data point is at the third stage of cancer or fourth stage of cancer, because the GLU activation function was actually employed, and the results that came out of this particular transformer model was yeah, results that came out of this transformer model after you see after softmax layer and linear function, the the output probability was around zero point 0.6 or so point 0.5 or like that. So I hope that makes sense.
Yes, yes.
Yeah. So to get a more continuous and more reliable output is why they use this complicated formula. But if you're asking me why half, then okay, like let's take it this way, right? If you use x equal to 0, you will have e power 0, which is 1, 1 plus 1, 2, 2 by 2, 1. So activation layer, I mean, phi of x starts at, but f of x is x into f of x. So it starts at 0, but at infinity, like since this is a continuous function, I'm taking infinity. So e power infinity is how much actually? Partha, do you have any idea? Hello, Partha, are you there? Okay. Okay. Anyways, we will look into this later. Yeah. Okay. So now we will get into the ESM fold and its working principle. Uh, yeah. So this is the bread and butter of today's, today's webinar, where we are diving deep into ESM fold. So ESM fold demonstrates the architecture uh, where we could utilize it for protein structure prediction. So the input sequence would be like a single protein sequence. It would take in the format of a FASTA sequence, and you do not. There's no need of multi-sequence alignment or evolutionary history. Uh, and when once it passes through the ESM2, the core of ESM2 is that it's a pre-trained masked language model that has learned protein grammar from millions of sequences. So it's able to predict residue-residue interaction based on contextual embeddings, much like how LLMs understand languages, right? So then we have the 48 block folding trunk. So once it passes through the ESM2, we have the 48 block folding trunk, where the transformer processes the sequences through multiple layers of 48 blocks to extract the structural information. So once this amino acid sequence is passed, here you can see, that's my mouse not working. Uh, yeah, as you can see here, uh, this this step, where it is passing through the transformer model, process the sequences in 48 blocks. So you see whatever the single sequence that you can see, this folding trunk, it is 48 blocks, and it extracts the structural information. Now this step is analogous to how AlphaFold refines protein conformation, but without requiring evolutionary alignment. And then we have the structural module, which is an 8 block structure which recycles the protein structure. So the final structure refinement happens in this module using 8 additional blocks, right? So the model also includes a recycling mechanism, meaning the structures iteratively improve for higher accuracy. And then it predicts the protein structures with the confidence score. So the final output is a predicted 3D protein structure with an associated confidence score. This allows researchers to assess the prediction reliability for experimental validation. And later, after predicting the protein structure, it recycles again where it goes to the 48 block folding trunk. So why it does that is that because it processes the sequences through to extract structural information. So any missed out structural information that is predicted from the first iteration can be recycled by passing through the folding trunk again, and then this is like an iterative loop until it zeroes down on a certain protein structure. So this is how the ESM fold works. If you had attended my seminar that I had talked about, like on uh, two couple of months ago, I had talked about how RF diffusion uses the concept of diffusion to predict novel proteins. ESM fold uses this particular block architecture to predict the new structure with new confidence scores. Now we dive deep into the aspect of confidence scores, right? So before that, I'll run you down the differences between the comparison table between ESM fold, AlphaFold, and Rosetta fold. So this table provides a detailed comparison between ESM fold, AlphaFold, and Rosetta fold, which are the three leading protein structure prediction models. Each of them have distinct advantages based on speed, accuracy, input requirements, and best use cases. So as I've mentioned before, ESM Fold was developed by Meta AI, focusing on fast protein structure predictions. AlphaFold was created by, this very well known, renowned scientist named Daniel Hassabis, right? So Hassabis created Google's DeepMind in order to create tools like AlphaFold, which revolutionized the field of structural biology by achieving near experimental accuracy. So the accuracies that were predicted of these protein structures using AlphaFold was quite top notch, which is why they have utilized. They have been scientists all across the globe have been relying on AlphaFold for protein structure predictions. Then we have Rosetta Fold, which was actually developed by Baker Lab. Baker Lab, which is a lab in University of Washington, they designed this multi-protein interaction modeling structure, which contains a transformer-based large language model. So sorry, not Rosetta Fold. Rosetta Fold does not contain transform-based model, but Rosetta Fold utilizes the concept of diffusion to find out novel proteins. And the model type. So model type of ESM fold is like transformer based large language model. So it learns protein structures based on cell supervised learning. So there is no new data set fed. It is able to supervise itself to learn from these protein structures, from like previously existing vast protein sequences. So there are resources like UniProt, SwissProt, and KVProt where you're able to just where you can find like protein sequences that repositories. These are predominantly protein sequence repositories that can be accessed from these websites. And this is the this like transformer based models learns protein structures from vast protein sequences. So there's no need of reliance on multi-sequence alignment or evolutionary data. AlphaFold is just like an all-in one house of a transformer plus multi-sequence alignment architecture. So ESMFold is just a transformer based, but AlphaFold caters to, the difference is AlphaFold caters to multiple sequences. So in the same time. So ESMFold only focuses on one protein sequence, which I'll show you in the next upcoming slide. How does ESMFold predict protein structures, right? Whereas Rosetta Fold is like a three-track network. So it does. So by three-track, it means that it integrates sequence, structure, and evolutionary data. So it traces back to the evolutionary patterns of that particular protein sequence, and it's not just used for like structure prediction, but it also traces your ancestors, the ancestors of the certain protein. So Rosetta Fold is basically designed for predicting multi-chain protein complexes and interactions. Input type is just like I mentioned, ESM fold is just single sequence, whereas alpha fold is multiple sequences alignment plus evolutionary data. And Rosetta fold is MSA plus structural templates. So structural templates which combine sequences and known structures for better prediction. In terms of speed, ESM fold is lightning fast. Alpha fold is slow. Rosetta fold is faster than alpha fold, but not faster than ESM fold. So accuracy, ASM fold comes about 85%, but what stands out for AlphaFold is that it is highly accurate. You can highly rely on AlphaFold's predictions on a certain protein sequence, whereas Rosetta Fold is around just 90% of AlphaFold's accuracy. Uh, so the best use cases for ESM fold is for large scale protein screening. So if a particular protein needs to be focused on in greater depth, then you can use ESM fold. But if you want to use for accurate structural predictions, then you can use AlphaFold. And if you want to use for a quick way of modeling and protein interaction, then you can use Rosetta Fold. In summary, if you're going for speed, then you should definitely use ESM fold. But if you want reliable, accurate proteins, then you have to use AlphaFold. And if you're looking for, like multi-protein interaction, like if you want to know what if this protein could interact with some other protein that that was not previously thought of, if you want to experiment with a lot of combinations of proteins to find a novel drug, if you're getting into drug discovery, then you can use Rosetta Fold because it is better used for multi-protein interaction and complex modeling, like it's three-track network architecture and protein-protein interaction. So because it's being a little faster than AlphaFold. All right. So next we will look into the applications of ESM fold in bioinformatics. So by utilizing a single sequence as input like this that I'm showing right now, this is a prototype of a P53 protein structure. So P53 protein structure is like the causative agent of cancer. So take this protein sequence as an example. I am going to show how a single sequence input greatly speeds up the prediction process while maintaining high accuracy in predicting atomic resolution structures. Moreover, ESM fold outperforms other methods when dealing with proteins that have few homologous sequences. Some key applications of ESM fold include metagenomic protein structure prediction, So it predicts structures for unknown proteins in environment samples. It helps discover new enzyme families and microbial functions, and it is a rapid structural analysis of large protein data sets. So it enables high throughput structure prediction for thousands of proteins, accelerates function insights for drug discovery in genomics. Then we have protein evolution studies. Reconstructs ancestral protein structures to study evolutionary changes, then aids in understanding structural conservation and divergence. Then we have drug discovery and protein engineering. Assists in designing stable proteins and optimizing drug targets. Identifies novel conformations for druggable proteins, and then we have function annotation of unknown proteins. This predicts structures to infer functions for uncharacterized proteins, right? So if there are some uncharacterized unknown protein, it predicts the structures that for that particular protein. And it also supports AI-driven annotation by matching these protein structures to like known domains. So if a protein is left unclassified, it is able to classify that protein and what is that, like it is able to um, move it to or yeah, it is able to classify that particular protein in a certain and match it to a certain domain. Yeah, match it to a certain domain. Next, okay, now we look at uh, how this is a simulation of the metagenomic atlas of ESM4. So if you go to the official metagenomic atlas of ESM4, this is how it looks like. So there's a huge globular structure of proteins. So this is the metagenomic atlas of protein structure. Guys, this is very interesting when it comes to understanding proteins, because this is a visual representation of how a protein sequence looks like. So when you click on that particular protein, or if you want to zoom in on that particular protein within the metagenomic atlas, you are able to you can download it in the form of a pdb format, which is a protein data bank format, or a sequence format. So you will get it in the form of a FASTA sequence or an amino acid sequence. And you have different alignment metrics that include predicted alignment and error. So not all of these proteins are accurate, right? And to so what this ESM4 metagenomic atlas shows is that you could also let me replay it for you again. So here is the globular structure of the metagenomic atlas, and these are based on coordinates. The on the right corner that you see, these are the coordinate numbers. So when you go to if you get the access of a coordinate number from a repository like Uniprot or KBprot, you're able to you can input that particular coordinate, and you will be able to see the protein structure. And once the protein structure is found, you can download it in the form of a pdb file or a sequence, and you can find the pldtt score. So pldtt score is something that I'm going to talk about in the next slide, which is really essential when it comes to understanding protein folding, because it tells you about the residual proteins that may not be highly accurate to the original protein structure, and it allows you to understand which protein sequences needs to be binded with which protein sequence, right? So what so this ESM fold is a high speed alternative to AlphaFold, but it comes with cost. Its accuracy is less. uh, yeah, wait one sec. Yeah. So once you download the protein sequence from the model, and when you can perform a protein prediction function like this. So here I'm downloading I'm downloading libraries like Torch and ESM, which allows you to um, uh, perform the pre-training aspect of this model. So what happens with this is that, like this is how a .pdb format looks like. The it can be used. I mean, it can be viewed using one or several free and open source software tools like Jmol, PyMol, PMD, and Rasmol, and other non-sharing shareware programs Okay, so like UCSF, which is UCSF Chimera, which is devised by scientists from University of California, San Francisco, uh, and Swiss Prot, PDB Viewer, Star Biochem. All of these are Java-based interactive molecular viewing platforms with integrated search of protein data runs. And all of these are quintessential. So in this particular case, we are inputting a protein sequence like this, and we are getting an output, right? So once we write the output, we get we get predictions like this. So what can we understand from these predictions as an AI scientist, or as a drug discovery scientist? uh, Once you get an output like this, what to how to make it head or tail about these uh, predictions, right? So I'll run you down. So this is how a general PDB file format looks like. So the described PDB file may be viewed using one of like it can be viewed using JMOL, PyMol, VMD, or RASMol. And the L that you see here, the L that you see here is nothing but light chain. So there are there are heavy chains and light chain when it comes to amino acid sequences. In this case, we are looking into, in this particular example, the subject is looking into aspartame, which is an amino acid. So to see how this chain interacts with the protein, uh, like we can classify it as a light chain with sequence number. So in this case, all across this rows, all across these rows, the sequence numbers mentioned as one, which means that it's printing out the first sequence in the amino acid sequence. In this case, it is aspartic acid. So what you see here is aspartic acid. So within this amino acid sequence, this N, C, A, C, O, aspartic acid is the first amino acid sequence that is being printed. And these are the XYZ coordinates of that protein structure. So, so it is pointing out to the first sequence in the L chain, and this XYZ coordinates shows the configuration of that protein sequence. So basically, if you're mapping this on a 3D visualization platform, you'll be able to view the protein sequence. Say, so this XYZ coordinate that you see here is the coordinate uh, that can be viewed from this metagenomic atlas cloud, right? You click on that protein sequence, that is the particular coordinate of that protein in terms of 3 dimensions. So when you download the PDB sequence, you will understand that this XYZ coordinate is the coordinate of that particular atom, which contains this particular element of this amino acid sequence. So it is very fascinating how we can interpret ESM4 predictions to useful ways to understand proteins. And later on, we will dive into TM score versus PL DDT score. So TM score, right? In bioinformatics, TM score is nothing but template modeling score. These are two essential metrics that we need to understand as a bioinformatician when it comes to assessing the similarity between two proteins. So when you're superimposing two proteins, how do you know that this protein is similar to the other one? So that is the underlying what is that underlying principle that every drug discovery scientist or a bioinformatics person would get into. And TM score and PLDDT score are two metrics that validates that claim. So TM score you assesses the similarity between two proteins. So when you're getting, so R is equal to 0.52. So in this case, what here the PLDT score and TM score, we are creating a correlation plot between them. So if it is ranging between from 0 to 1, where 1 signifies a perfect match. So so you can see like this protein, like you see this data point here, this tiny protein data point here, it is almost close to 1, where TM score is 1 and the PLDT score is 1, which means that that particular protein sequence is very similar to the reference protein sequence. So the Pearson correlation between the TM score and PLDT score, in hindsight, by taking the average of all of these data points, it's showing as 0.52, which means that the score is pretty low in terms of the correlation. It's pretty low, which means that this is a low quality metric that cannot be used to distinguish between alternate conformation. So you cannot distinguish between alternate conformation with this particular score. So um, like color sequences encoded across each strand of these protein uh, has specific color demarcation that indicates the PLDDT level. So if the PLDDT level is between 0 to 50, which is denoted by orange, it means that that protein structure, like you see in this protein structure, orange is depicted here, right? So the distance between of that particular, it's called residue from of the protein sequence. Each strand of this protein sequence as a whole is called a residue. So the distance between that and the original protein sequence is low. I mean, is high. So which is why the PLDTT score is less. So more the distance is, the further it is away from further it is away from the original protein structure. So PLDT score is not the accuracy of prediction, since it does not compare with like experimentally solved structures which were run through like X-ray crystallography or cryo. It only estimates the reliability within the model's own framework. So here you can see that the PLDT score is 0 to 50, whereas at some protein structures like dark blue here is 90 to 100. So, and most of them are like light blue, which means that it lies between the range of 70 to 90. So this is the PLDT score that suggests that this protein structure cannot be highly reliable, because most of them are light blue, which comes under the category of being moderately reliable, where the score is like around 70 to 90. The more it gravitates towards 90 to 100, the more reliable it can. So this is a parameter that is used while looking at the structural reliability of protein structures by drug discovery scientists and AI scientists alike. All right. So yeah, this is a graphical representation or a video on how these PLDT scores look like. So like I mentioned, if dark blue is very high, so PLDT score will be above 90 at that particular place. Light blue is about 90 to between 90 to 70. So PLDT scores is a score that does not rely on superposition but assesses the correctness of the local distance. On this basis, a PLDT score above 90 would be taken as the highest accuracy category, in which both the backbone and side chains are typically predicted with high accuracy. In contrast, a PLDT score between 70, above 70 corresponds to a correct backbone prediction with misplacements of side chains. PLDT tells you where to trust the 3D model, whether you have to trust this or not. Low PLDT regions suggest potential disorder, dynamic behavior, or binding induced changes. There might be so many changes within the model that can be disregarded, where that may not be of much importance. Whereas high PLDT score matches well-defined 3D elements with like helices and sheaves. So high PLDT scores can basically match well-defined 3D structures. So like this portion, this dark blue portion that you see here in this video, if you're binding a protein sequence with a similar like confidence score, then it is showing that matches with the well-defined 3D elements of that particular protein sequence. Now we will dive into the case study aspect. Okay, so I'm going to show you a code demonstration and how this particular model. Wait 1 second. Where's my hello? Yeah. Okay. So Sorry, 1 second. 1 second. I wanted to show you the code.
Mrinal, also a time check.
Yeah, yeah, it's ending. It's towards the
Okay,
end. So
okay,
yeah, I
thank
just wanted to show you the case studies, but I don't know why my laptop has gotten frozen for some reason. Uh, so do you know any? I don't know why it got frozen. Sir, am I audible?
Yes, absolutely.
1 second. Show all windows. Okay. I'm going to open it on a new window just to show you. Why is this getting stuck? Mm, Um, I don't know why my laptop is getting stuck.
You want to reboot and rejoin?
Is it okay? Like, can you give me like 5 minutes? I'll rejoin that.
No, no, 5 minutes is a lot. Will it take
Okay,
that
two
long
minutes.
to
2 minutes.
reboot?
Just give me 2 minutes. I'll be back.
Okay,
Yeah.
okay, fine.
Yeah. Let me try now.
So, folks, meanwhile, let me apprise you of our activities in Shellside. So, we had a good healthcare hackathon, on 8th of March with the 100 teams registering, and finally 25, participated. So, we are meeting these teams, the 25 finalists, again today in an online workshop to see if we can guide them further in their projects. And we are planning other, healthcare hackathons in coming weeks and months. So, idea is to accelerate the healthcare research and try to do some good innovations. Let's see. If any of you are having extensive, deep expertise in healthcare, reach out to us. We'd like to collaborate. So, is anybody here involved in healthcare, projects or healthcare tech or healthcare AI? Okay, I guess not. So, So we are interested in developing some interesting apps in GenAI for healthcare and connecting with the community doctors and hospitals, medical colleges slowly to create an ecosystem. Just like we have a AI tech community, we would like to probably engage the healthcare community. So that's the goal, and see if we can, working in collaboration with them, produce some deeper apps. Because when engineering teams develop healthcare projects in hackathons, they don't have domain expertise. So it is critical to get the domain experts to guide the healthcare innovations. So that's why we are reaching out to medical experts, doctors, hospitals in India so far, in Bangalore particularly, but over time expanding across India and globally. Feel free to share any other thoughts. I hope you are enjoying the Selstrat webinars, and courses. So we have done recently an agentic AI course, which was well received with huge attendance, and we are planning to launch further, courses in agentic frameworks and so on. So just a show of hands, is everybody here from India, or do we have folks calling in from abroad as well? Okay, Brinal is back. Brinal, are you ready? You're on mute, Brinal.
Am I audible, guys?
Yes, now you're audible.
Okay. Yeah, so
So
sorry for that.
share your screen.
Yeah. Uh, yes. Can you see my screen now?
Yes.
All right. So yeah, so now we are going to look into the case study of today's class, today's module. So basically, uh, let me just open that slide. Yes. So we are going to, yes, the case study part of this module, right? And we are going to predict a disease-related protein structure. So I'm going to run you down the code. I have the protein structure, all the results stored already because I ran the code previously. But I'm going to run you down the code so that you can have a brief understanding of what goes into each aspect of this code, right? So I'm installing these essential packages like ESM OpenFold Y. So ESM fold, you can do a pip install, and this is a particular Git, repository. So like it is already found on GitHub, so you can install it from Git. Then this is an essential series of what would I like to call it? These are the essential series of models that needs to be installed before starting the designing of the protein structure. So it includes BioPython, which looks into the BioPython package is basically for bioinformatics purposes. Py3D-mol is to view the so you have these molecular structures in and you can view them in various, like I mentioned, like various tools like UCSF, Chimera, PyMol, and all of that. So to view them in a 3D format, you need to use packages like Py3D-mol. Transformers is also needed, Torch, and Request. So all of these three models are required, and then yeah. So I have uninstalled it after I. So please don't follow this command. This uninstallation was done after I perform the particular what is that function. And so here we are doing, this is the most important aspect of the code where we are predicting the pre-structure of the protein sequence. So in this case, I am loading the auto tokenizer ESM4 protein folding from the transformers. And there's something called SwissProt, and these are library packages within Bio. The reason why I'm installing Swissprot and Uniprot is because like Swissprot contains all of the databases information that is present in this um, particular uh, repository called Uniprot, which if I'm going to show you, which would look like this. So I just looked up. So Uniprot's official website looks like this, where you can type in the accession ID number of that protein sequence, and you're able to search through that protein sequence. So Uniprot's, like this, or the I've almost performed a web scraping kind of function where I don't have to essentially go to the Uniprot database, but I can access the Uniprot database just from my block of code. So this is why I've used Swissprot, because it contains packages like it contains functionalities like read and like sequence and all that to get the specific Uniprot ID from the package and then return the sequence. So once the sequence, so say you're passing, so here I'm doing something called cleaning the sequence. So this is a pre-processing step that I'm utilizing, where so in an amino acid sequence, you will only find characters or sequences within the within these alphabets. So like from A to Z, I have mentioned only the alphabets that include amino acid sequences. Like, say, if you can see here, B does not exist here. So if you count through this, it's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. Yeah. So there are only 20 sequences, right? So you remember I mentioned at the start of the module that there are only 20 amino acid sequences in the human body architecture. This correlates to that, where you can see that the starting letter does not, like, if you correlate this code with this particular image, you can see that you don't have protein sequences starting with B. Can you find a protein sequence that's starting with B here? There are no protein sequences that's starting with B. So hence we are performing pre-processing by utilizing this particular string. Another example maybe A, B, C, D, E, F, G, H, I, J. Okay, let's see if there are any protein sequences with the name J, with the starting letter J. There are no protein sequences with the starting letter J. So so I have created a pre-processing step where it cleans the unwanted amino acid sequences. So this particular function was performed as a pre-processing step, and once the protein is being pre-processed, now I pass it to the predict structure function, where it predicts the 3D structure of that function. So this is the most essential important part. So what I'm doing here is I'm running torch.device uh, on CUDA. So CUDA is basically using the GPU server. So in my Google Colab right now, it's running on system RAM and disk. But once I run this function, it will connect to my GPU server within Google Colab. So once it gets connected, it will either
Yeah, we
use.
can increase the font a little bit.
Sure, sure, sure. How about this? Is this legible? Is this
Bit more.
bit more? Okay.
Yeah, this is good.
Okay. Okay, so now here I am running it within Google Colab, Google Colab's GPU server. This particular line does that, and I'm calling the ESM for protein fold function that I've imported from Transformers model. So this ESM for protein folding function loads the tokenizer, which is part of Meta's. So Meta has designed ESM fold, right? So it'll load the whole ESM fold version 1, and it'll mount it to the GPU here. So once it's mounted, what it does is that it will uh, perform. So if there are torch no graph, so which means that it'll this particular function makes sure that there are no what is that, gradients, or there are no residuals in the particular protein structure. If there are no residuals, then it will perform the operation where it will infer the PDB of that particular sequence. So inferring the PDB step does nothing but it infers the protein information of that particular protein sequence. So when you're performing a no-grad function, what a no-grad function basically does is that it basically what it does is that it removes all the residuals that are in the particular protein sequence. It removes all the underlying residuals, and you are able to get a fully formed structure of that protein sequence, fully reliable protein structure, right? And now we are saving this protein structure. This is our last step where we are saving the protein structure in a PDB file format where it can be viewed. Right now, I'm using you can use multiple tools. There are JMOL, there's PyMol, there's UCSF Camera, but I have installed PyMol, and PyMol I think is the most basic tool for a starting of drug discovery scientist or someone who's interested in understanding the functioning of protein molecules. They can install PyMol, and this will be saved in the format of a PDB file. So now this is the main execution step where I'm just passing in the UniProt ID of this particular protein, and then I'm fetching the UniProt sequence, then predicting structure and saving the PDB file. So it does all of this operation, and then finally okay. So this step I save it in the form of a UniProt ID slash structure dot PDB. So whatever you so say I'm typing the same thing on UniProt and searching for that particular protein P0436, right? So P0436 7, I think 7 is also there, right? Yeah. So yes, so this is the protein sequence, and it is an anti-freeze protein that lowers the blood freezing point. Okay, so if I want to view this protein sequence, I can view it from UniProt too. It looks like this. So the protein structure looks like this, but you don't have what you call like residues to know the PLDT score that I mentioned. It is there, but it is not so prevalent. You can see the 3D model of that protein sequence, but you don't know where the target binders, which part of the protein sequence does the protein, does the drug bind to. You don't have these information, and moreover, it's not very clear. When you zoom in, it's blurring a lot of the images, which is why we use something called PyMol. So I'll show you how the PyMol looks like. Sorry for this. So PyMol is nothing but a software tool that is used to visualize protein structures. So if I were to PDB, yeah, okay. So I'm opening this particular file, molecule. One second, I have a lot of tabs open. Um, yeah, I have, where's the .pdb file? Let me just open PyMol first. So PyMol is this particular visualization tool for the protein sequence. And yeah, so yeah, this is the structure. So I'm going to open structure PSC1. Okay, yeah. So structure PSC1, right? This is the same protein structure that you're able to see on PyMol, you're able to see here as well. So this protein structure, which is less clear on the UniProt database, see, you can see the blurring happening, even though you're able to see it's not that evident. But through PyMol, you're able to see the intricacies, the the this is, you see the arrow marks here. This shows the arrow marks of how the hexical binding is happening within that protein structure. So basically, these arrow marks denote how this folding is happening. So all of this can be viewed from PyMol, but Okay. So this is the predicted ESM fold. Uh, so this is what ESM fold predicts this protein to look like, right? This is the output that we get after performing this step. Now here I'm going to compare between two protein structures, which is from AlphaFold and uh, what I got from ESM fold. So the same file, the same Uniprot ID file, I am loading it from a different website, that is AlphaFold. AlphaFold is also having a server, as having a repository where you can search for the particular protein sequence. And uh, so once I download the file, I have saved it as structure uh, psc.2, right? Right. So this is the output that I get of the same molecular structure uh, AlphaFold. So whatever you see here is the output. Sorry for this. Is the output that you get on AlphaFold, and the stark differences here. You can see there are some differences here. This shows the um, binding targets of that particular protein sequence, whereas, okay, sorry, I have open structure, yeah, sorry. And here you can see there are different colors associated, right? You can see the light blue color, whereas here this is a plain protein sequence. So AlphaFold has a more nuanced nuanced and more accurate finding. I mean, not yeah, accurate finding. I can say this with confidence because uh, these colors represent the PLDTT score that I mentioned previously. The PLDTT score, that means that if if it is of light blue color, um, like you can see here, if it is of light blue color, it falls under the category of 70 to 90. So 70 to 90 meaning that these proteins, this particular structure of that protein is highly is can be relied upon, can be relied upon, and can be taken, can be chosen as a target for the drug molecule to bind and see for predicted results. Now the next step would be passing in a lot of smile strings of that particular drug molecule and trying to attach it on these binding sites. So this particular point of that particular protein is a binding site of P0. What is that? What is the protein called? P04. Yeah, P04637. So this is the stark difference of what an AlphaFold PDB prediction would look like as compared to the PyMol. As compared to the ESM folds. Now I'm going to compute the RMSD distance. So RMSD distance is nothing but the root mean square division distance. So what RMSD distance does is that you're importing these two structures and you're calculating the distance between the proteins. So it superimposes one protein over the other and it calculates the distance between these two proteins. So when it does that, it calculates. So a typical RMSD score of a it calculates the similarity, so we can check which output is more reliable. Is it the is it the AlphaFold's one or ESM Fold's one? And usually when you look at RMSD scores, you get an RMSD score for AlphaFold and ESM Fold, uh, like AlphaFold median backbone prediction, and so say you have a RMSE score of around 0.96 Armstrong. Armstrong is the uh, is the unit that you use for like the calculation of the size of a protein. If you're getting between of PLDT score of 0 to 5, it indicates a high confidence. So for this particular protein, I got, like, when I tried the RMS, when I implemented this function, I got around 2. So I got value around 2. Now it's not showing up. But if you run this Colab notebook of mine, you will be able to find out in terms of Armstrong how much is the RMSE distance score, and I got it around 2, which means that it can be relied upon. So this particular protein structure is almost similar to this one. there might be just 2 Armstrong differences between them. So so a PLDT score of sorry, an RMST score of around let me just ask RMST score reliability. How much? So if it is between the range of 55 to 60, it is acceptable. So so this is what the internet has to say about RMST. So if an RMST less than 2 corresponds to molecular docking solutions. So molecular docking is a concept where we are superimposing a particular molecule to another molecule, and in this case we are trying to bind. So the further steps of this would be trying to bind a protein molecule, or I mean drug molecule, onto this the protein molecule. So you would say, I mean, when you run my notebook, you will get around 2. I think I got 2 as my 2 Armstrong as my value. So which means that my prediction is almost accurate. So yeah, this is what I wanted to show you as a case study for today. And if you would like to run this and see the results for yourself, please feel free to reach out to me. I'm always there to give you the Google Colab notebook. You can run it. And yeah, that is a bit about the case study. Now the final conclusion of this whole first module series, I'm going to talk about the inference of these results, right? So in the inference part, I'm looking at the key evaluation steps. So evaluation of AI models. So you have key evaluation metrics like the RMST evaluation and TM score evaluation. So I had mentioned about this, right? So so in the you can see in this graph, sorry, in this graph, you can see that there is, sorry, let me just close this. Yeah. So you can see that there is, like bandwidths of, there are number of data lines here. So what can you infer from this graph? This graph shows how the scores for each protein sequences are differing across time, right? So from 0 meaning no similarity to 1 being perfect match, uh, in this RMST evaluation part, what you can infer is that it measures the average distances between atoms of predicted and experimental structures. So lower RMST means higher structural similarity, sensitive to outliers and global alignment. So when you see here, the red trend line shows a high similarity, which is going from zero point 0.3 to zero point 0.6, whereas a black one, which is of 298.15 K, it is showing from point, there's very less deviation, whereas this protein, whichever protein that is actually fitting to that particular reference protein better. So you can say that the red trend line is highly reliable when it comes to, through this particular graph, you can say that this red trend line of that particular protein sequence is highly reliable. Now, if you look at TM score evaluation, so in TM score evaluation, as you can see, this is a combination of 20 protein sequences. These are the names of different proteins list, where you have Z scores above 2. And you can see here that, 0 means no similarity to 1 means perfect match. Now, about TM score above zero point 0.5 is often considered correct fold, right? So here in this case, you have TM fold above zero point 0.5 for most of these proteins. The ones that are in yellow are most highly reliable, I think, because they they contain um, they contain the scores that indicates correct fold. So anything above zero point 0.5 is considered reliable, and the yellow protein, the proteins that belong to the yellow family would be considered most reliable, because most of them are above zero point 0.5, and they have two protein structures that are around 0.61 and 0.610. So if you're taking a cumulative comparison, the proteins that belong to the yellow family are considered more reliable to a certain protein structure. So yeah, benchmark data sets, we have, like I mentioned, PDB, like we downloaded the file in the form of a PDB format, and we have CASP16. CASP16 is like it's like a gold standard repository for experimentally determined protein structures. It acts as a reference data set for model benchmarking and evaluation. So for evaluation and model benchmarking, we use CASP16. Yeah, then application of AI powered protein folding, it is applied. So see, you see P53, this is the cancer causing protein. This is the 3D structure of the cancer causing protein. And this protein can be assessed through formats of like drug discovery, for drug discovery scientists to look upon, or enzyme engineering or synthetic biology. Um, and this accelerates rational drug design, enabling pharmaceutical companies to screen molecules and optimize hits faster. So, for example, cancer therapeutics targeting P53 and enzyme engineering. So, AI-powered prediction speed up synthetic biology applications where custom enzymes or pathways are designed for specific tasks. And another one is vaccine development. So, accurate structural predictions help identify antigenic sites. So like for P53 protein. So if you're using it for vaccine development, say for COVID, not P53 in general, but any other protein sequence for to prevent a pandemic or an epidemic to happen, we can rely on AI powered protein folding methods to find out, um, identify antigenic sites and that are crucial for designing vaccines. See, for example, I'll give you an example. So COVID-19 happened, right? mRNA COVID-19 vaccines leverage spike proteins. So you in this GIF, you can see the green ones are those spike proteins. So mRNA COVID vaccine leverage the spike protein structure to design the immunogenic region. So to find out which part of our body are most immune to COVID-19, the spike protein structures were more analyzed as amongst the flowery structure of that protein, right? So this allows faster response to emerging infectious diseases. So all of this can be achieved using AI and AI forms of protein folding. So I believe that's almost conclusion of uh, this presentation. And finally, the future directions of AI and protein folding. So I think I've discussed from 2000s to 2018 what all have been happening, and also talked about the breakthroughs. But what we can expect in the future is something really fascinating, because um, in we can we can expect things like this now. We can expect de novo AI assigned de novo proteins, where AI models will design new proteins not found in nature. There will be novel proteins that have not been discovered that can be found through AI, and potential for tailor-made proteins with therapeutic functions. So say for cancer killing, for neurodegenerative diseases, if you want to find novel proteins, you'll be able to find using AI and real-time protein structure predictions. All this while we were getting data sets from already pre-processed data that were in the medical repositories. But how about we create? So you would have heard yesterday there was a news about a 16-year-old boy who created an AI that could detect heart diseases within seconds, and he won I think a national prize. Yeah, Chief Minister of Telangana, I think Chandrababu Naidu, acknowledged this and innovation and gave him like a state prize or something like that. So, so that is one example of how AI has, if AI is is intervening in modern medicine, it can accelerate the accelerate the detection of diseases. Someone who can, a 16-year-old boy using AI, if he's able to create uh, a, a sophisticated system that is highly reliable and accurate, and that can detect heart diseases within 7 seconds, then don't you think there can be a lot of advancements that we can think about in terms of disease detection and drug discovery when AI comes into the picture? So these are multiple. These are some of the examples that I can think of that would come about in the future, that would be va- that will be revolutionizing the the field of drug discovery and protein folding and disease detection. So that's all I have for today's module for the introductory section or introduction to generative AI and bioinformatics. In the next modules, we'll be dealing, getting, diving deep into the different models, like I've mentioned in the course structure today morning. Like at the start, we will be looking into the genomic data analysis, the structural and functional proteomics. We'll be diving deep into the AI intervention in all of these bioinformatics concepts. I hope I have created an engaging content, and hopefully you've all, I hope I have, take not wasted your time, and hopefully you have learned something new today and something that can be taken to your, to your work sometime and being discussed upon within your colleagues and your loved ones. So hopefully there's some good takeaways from this module from today's presentation. And yeah, thank you, Vivek sir, for giving me this wonderful opportunity to showcase my whatever possibly less knowledge that I have in bioinformatics, to give me this platform to showcase my knowledge in this platform. Yeah, thank you very much.
Okay, good work. So a lot
Yeah.
of extensive stuff on the basics of bioinformatics, AlphaFold, ESM fold, etc. Audience, any final questions or comments?